a great pleasure to be here. You already see C. elegans in the background. And today I'll be talking about evolution of C. elegans microbe interactions. And I will present to you sort of a short overview of some of the things that we are doing. So in general, my group is interested in the evolution of host and microbes. We use C. elegans for most of our work. And I'll try to move it here. So we look at C. elegans pathogen coevolution. We are interested in the genetics of the interaction, which includes immunity. We're also actually one of the very few labs looking at the natural ecology of C. elegans. And in this context, also C. elegans microbiota interactions. And part of my lab is working on antibiotic resistance evolution, also doing a lot of evolution experiments. But this is something I'll be not talking on today. Kiel is a wonderful environment to do this kind of research because over the last decades, Kiel has emerged as one of the hotspots for evolutionary research in Germany, thanks to a variety of different research consortia that we could get funded. And we will soon also even move into a new building that is currently being built for evolutionary biologists in Kiel and northern Germany. C. elegans, as you may know, has become one of the main model organisms in life science research. And this is due to a variety of advantages. You can easily cross them, do genetics, you can do easily a variety of different experiments, simple genetic manipulation. What is super nice is that the nematode is transparent, so you can look into a living system and see what's happening inside such a system. It's also super nice that you can freeze the nematode. I mean, you can freeze any kind of organism, but this one also survives it. So you can actually, have a lot of mutants, you can freeze them and you do not need to maintain them with a lot of efforts. And in this way, C. elegans has become an important model, more or less across all of the biological disciplines. And what is fascinating for me with a background in evolution and ecology is that more or less all of the C. elegans research has been done in the absence of its natural microbiome. And this is due to a protocol that was introduced right from the beginning, which sterilized the nematodes. So all of nowadays we have more than 20,000 labs across the world working with C. elegans. They all work with a single strain. That's another funny curiosity of C. elegans research. And they all work with under sterile conditions, more or less, and with only a single food organism that the nematode never encounters in the wild. So this is a little bit strange, and you could argue that we may be missing something very important. And this is something that we also try to address in our research. What has been funny, when I started working with C. elegans, there was even not very good understanding of where the nematode lives in the wild. And this is where you can find them. The nematode loves rotting plant material, especially rotting fruit or rotting apples. So usually from September until the end of the year, you can actually find C. elegans in large abundance in orchards or in compost heaps. And based on the research of only a handful of groups, so even until today, there are only a few groups that are actually looking at the natural ecology of C. elegans. We do have a little bit of knowledge, but we also still have huge gaps in our understanding of the natural ecology of the nematode. So here you see an overview of some of the vectors that are relevant for C. elegans, competitors, predators, and we also have a little bit of an understanding of microbial interactors of C. elegans. But it's only the tip of the iceberg, so we still have large knowledge gaps. Why is this relevant? We pointed out 10 years ago in a review paper that a large part of the C. elegans genome is still not understood. So what you see here is all of the genes in the genome that have different kinds of annotations, and the black part is the important ones. These have been the genes in the genomes without any GO-term annotation. And on the right, the percentage of the genes without any kind of phenotype annotation. That was 10 years ago. We repeated that analysis last year. It's a much stronger reduction here regarding GO-term annotation, but phenotype annotation, we're still above 50% of the genes of the C. elegans genomes have no phenotype. And again, considering that more than 20,000 labs are working with this nematode, this is amazing. This is really strange. So C. elegans is one of the best studied organisms on Earth, and still we lack information about the majority of the genes. And in my understanding, this is due to the fact that we are ignoring the natural context of this nematode. So again, this is something we are trying to change with a variety of different approaches. One is, of course, based on experimental evolution, but also field studies. I'll be touching these three topics today. I'll briefly present to you some older work on host pathogen coevolution, then on the evolution of host-associated microbes. This is actually more focused on the microbes. And then I will talk a little bit about C. elegans microbiome interactions. So when we started this kind of work using experimental evolution with C. elegans, I was still mainly interested in host pathogen interactions or host parasite interactions. And we chose C. elegans as a model, and there were a variety of open questions at the time, including what are the exact selective dynamics of coevolution and what is actually the genomic basis of such coevolutionary interactions. And I briefly highlight that the two main hypotheses about the selection dynamics for host pathogen coevolution look like this. So either you have recurrent selective sweeps, so novel variants appear, they sweep to fixation, and then you get the next one at some time point. The alternative is frequency-dependent dynamics, where you actually have host and pathogen alleles that never fix, but only or always oscillate. So you have fluctuations across time of the same kind of alleles. And there had been a debate which of these dynamics are more important. They have been addressed in a variety of organisms, and we try to contribute to this field by doing experimental evolution with C. elegans. And I showed on the previous slide, this has been work of a former colleague, Andrei Papko. As a model, we chose the interaction of C. elegans with Bacillus thuringiensis. There are bacilli strain that very specifically infect nematodes. They do that via toxins that kill the nematode and break up cells, and that allows them the pathogen to invade tissue of the nematode and proliferate. And at the end of an infection, you see something like this. So here you actually have a corpse of C. elegans, and in red you see proliferating bacilli of this particular pathogen. So what we did is we performed a coevolution experiment where we had host and pathogen co-evolving with each other over 23 transfers, and we replicated this 16 times. And as controls, we had the host evolving under the same experimental conditions without the pathogen, and we also had the pathogen evolving under the same conditions without the host for the same time period. Then we took advantage of that we can freeze the material. So in regular time intervals, we froze everything for later genomic analysis, but also for later phenotypic analysis. And here we especially took advantage of doing time shift experiments, which I will explain in a minute a little bit more. And as already pointed out, so we also isolated DNA from different time points for genomic analysis of both the host and the pathogen. Time shift experiments are important for understanding the evolutionary dynamics of coevolution because they may actually already indicate what kind of selection dynamics are important. So what does it mean doing these time shift experiments? You take a focus antagonist, in this case a host, from transfer 10, then you expose it to pathogens from the same time point or from the past or from the future. And this is possible in the system because you can freeze both antagonists, and you can also do the reverse using a focal pathogen and exposing it to hosts from the past, the present, and the future. And the assumption is that when there are recurrent selective sweeps, we should see a pattern like this of the interaction phenotype. So going in one direction could also go up, but it means that the antagonist from the future behaves differently to the pathogen from the time now and to the past. So for example, the pathogen in the future is better able to infect the host, and therefore the host has actually reduced fitness because it adapted to over time via recurrent selective sweeps. When we're talking about fluctuating selection conditions, also called red-queen dynamics, we rather see a phenotypic pattern like this. So that contemporaneous antagonist is best adapted. And therefore, the host has lowest fitness, because it's the contemporaneous combination that is actually matching, or whether the two antagonists match each other. So doing this kind of analysis, we looked for these two alternative types of patterns. And when we looked at the host, we indeed overall saw a statistically significant effect of the second pattern. So a pattern that indicates red-queen dynamics, frequency-dependent selection. And I said we could actually, and this is one of the unique features of C. elegans, we could do the reverse thing as well, where we then focus on a focal pathogen and expose it to the host from the past, the present, and the future, and we got a similar signature. So very strongly indicating that indeed under these very specific selection conditions, we have frequency-dependent selection. What I found very impressive is that the scientist working with me at the time, Andrei, he wanted to make sure that this is really the case. So he repeated the entire experiment with more replicates, and he slightly shifted the focal time points. And over all of these replicates, including more technical replicates, he overall could recover again this pattern for frequency-dependent selection. And this is indeed the first time that this could be demonstrated, this kind of dynamics for a eukaryotic host organism for both antagonists. So previous work only focused on one of the antagonists to look at these patterns. And this was the first one where we were able to do that for both antagonists. So again, as a summary, very strong indication of frequency-dependent selection dynamics or red-queen dynamics. Then, of course, the next interesting question is what's happening at the genomic level. So what is underlying this pattern? So we obtained pooled population genome sequences from transfers 1, 12, and 22. In this case, we focused on 6 randomly chosen replicates. And the assumption is that the dynamics that we see across these independently evolving replicates is in parallel. And also as a reminder, so if it's negative frequency dependent selection, we would expect over time a pattern like this, or mirrored, so something like this. If it's recurrent selective sweeps, we would expect that something only goes up, but we may also see something like incomplete sweeps. And cutting a long story short, I want to show you more data. In the majority of cases across the genome, we saw a pattern of incomplete selective sweeps. So what you see here are a couple of different loci. Here you see the time points, here you see allele frequencies, in gray you see the controls, and in red you see the co-evolved host populations. And they usually go up during the first time period, and then they sort of remain at that frequency. So, and we could not find any locus where we saw a pattern of negative frequency dependent selection. So this suggested to us that we actually have different regions in the genome involved during different time periods. And this is, looking back, perhaps also not completely unexpected, because we know from functional genetic analysis that there's a larger variety of genes in the genome that are relevant for resistance against Bacillus thuringiensis. And it is very well possible that these then respond independently across different time periods. That is at least our interpretation of this data. And at least it is an important piece of information that the genomic data that we recover need not mirror the phenotypic data that is suggestive of certain selection dynamics. What about pathogen? The same kind of material that we analyzed, the same kind of expectations we had. And here it is important to point out that Bacillus thuringiensis has its core chromosome, a circular genome, that is illustrated here. And here we actually didn't find any kind of interesting pattern that matched the phenotypic dynamics. But this pathogen also has plasmids. And the relevant toxin genes for the interaction are encoded on plasmids. I'm only showing you one of the plasmids. Some of the strains of Bacillus thuringiensis can have up to 19 plasmids. And this is a story on its own, very fascinating what these plasmids are for. to a large extent, we still don't know. I'm highlighting this plasmid. This is a plasmid which encodes one of the toxin genes that was shown before to be relevant for the interaction with C. elegans. This is where the gene is located. And funnily, we obtained one variant during our evolution experiments where there was a large deletion of this region. We could use that variant to verify again that the presence of this toxin gene is actually important for the interaction. So here you see the Bacillus wild type. Here you see the one with this particular deletion. And here E. coli as a control. And here you see the number of dead C. elegans. So as soon as you have this deletion, then it's not killing anymore. So that was a nice confirmation that this gene also is relevant for our purposes. And then overall, what we saw regarding dynamics was a pattern that now did match negative frequency dependent selection dynamics. So what exactly do you see here? Here you see time, transfer time, and here you see copy number of this particular plasmid. And in gray, again, the control evolved pathogen lines, and in red, the co-evolving ones. And the co-evolving ones indeed show a pattern that is consistent with negative frequency dependent selection dynamics. And we did slightly other analyses that again confirmed that it is really the copy number variation that seems to be important for the coevolutionary dynamics that we observed. This is also very interesting because the coevolutionary responses happened really fast. And it is believed that very fast responses can be or may be more easily mediated by copy number variation. And especially in bacteria, this has been hypothesized and shown in different contexts that this is something that can be achieved via plasmids, but also via other mobile genome regions in the core chromosome. And this is a very nice confirmation for that rapid responses may often be mediated by such common copy number variations. So with that, I'd like to close this part of my talk. So we could demonstrate that there is temporal and reciprocal co-adaptation of host and pathogen that is consistent with negative frequency dependent selection dynamics. And the underlying genomics do not confirm that for the host. Here we conclude that it's different genome regions that are actually responsible at different time periods for mediating or for generating these dynamics, while at the pathogen level, it's plasmid copy number variation that is underlying the negative frequency dependence. With that, I'd like to move from host-pathogen coevolution to more complex interactions, the interaction between C. elegans with its microbiome. And this has been the focus of my group during the last couple of years, and we tried to look at that from a variety of different angles. We studied the underlying genetics of interactions with specific microbiota members. We did some field work. I'll show you a slide in a minute on that. And then we did experimental evolution, and that's something I will focus on right now. What you see in the background is C. elegans colonized with a member of its microbiome, Pseudomonas fluorescens, that especially accumulates at the beginning of the gut and in the hindgut region. Yeah, and I mentioned that we also, we did and are still doing fieldwork with C. elegans. And again, this is really fascinating because the natural ecology still is so unknown. We still have an enormous knowledge gap of where the nematode lives when. We still have no idea where the nematode is from January until end of August. If you know, please let me know. We have no idea, even though so we find this nematode on rotting fruits, but also compost, and the compost is always there, but C. elegans isn't. So very strange, very, very weird. But we are making progress also regarding interactors and different microbes and their relevance. And what I should point out, and I will come back to that a little bit in a minute, the nematodes like it if the apples are really, really rotten. So any kind of apple that you would not like to touch. That's what the nematode likes. So We now use the system, the interaction of C. elegans with sort of microbiome members, to again address a variety of different questions that are open questions, that are questions that have not been fully answered yet. And when I started my work in the context of host microbiome interactions, I was fascinated that most of the work has very much focused on the host. And as some of you may know, research on host microbiome interaction really has exploded only over the last 2 decades. Before, there was hardly any work in that direction, and it exploded because of advances in sequencing technology and the realization that the microbiome is so diverse and is so abundant, and subsequent data that then revealed that the microbiome is influencing almost any trait of the host, and is therefore really, really important. And as some of you may have noticed, this kind of research is currently revolutionizing our understanding of biology. But it's still fascinating that usually we take the host perspective. It's perhaps not unexpected because we are hosts ourselves. But we may be missing something important. Yeah, so we make the implicit assumption that the host is a control and the microbiota somehow benefit the host, and the host immune system controls presence of microbes. But we may be missing something important because the microbes have a much shorter generation time. They show usually a much higher evolutionary potential, so they should actually be in control of us. And this has not really been studied a lot. And this is where we thought we could contribute using experimental evolution. And to further address that, I should also point out what the challenges are for a microbe. And when we think about microbial lifestyles, then microbes may be sort of living somewhere in the environment, so they have a free living lifestyle. Or the other extreme is that they're exclusively living with a host. So they only are found in association with the host organisms. And we know some of these extreme examples that are symbionts that also become intracellular and are never found outside of a host. But what also often has been neglected and overlooked is that the very large majority of microbes have a biphasic life cycle. They do not exclusively live inside of the host, but they have part of the life cycle outside of the host. And this is true for a very large variety of microbiota members. And we also postulate that this definitely was the ancestral state for becoming host associated. And therefore, we decided to look at this biphasic life cycle in more detail and try to understand how microbes optimize their fitness. So how they adapt to this biphasic life cycle that we postulated depends on adaptation to different steps across this life cycle. So these microbes need to optimize life in the environment. They need to get into a host. They need to be persistent, be maintained in a host, and they need to get out again to complete the life cycle. So based on this, we postulated that microbes need to optimize their fitness across all of these different steps and not only one of the steps. We even used this kind of insight then to join forces with mathematicians and define sort of a mathematical model that then allows us to highlight microbial fitness. And this is analogous to what has been done before to parasite and pathogen fitness. And that used to be a game changer in our understanding of pathogens, pathogen evolution, parasite evolution, and is still actually a model that is used for understanding the epidemiology of infectious disease. As you may still remember from the beginning of the corona times, when everybody spoke about R0 of the coronavirus. To threaten you, we've defined an R0 for microbiome bacteria, but mainly to actually emphasize and highlight that what are components that are important for fitness of such microbes. So I will not go into detail much more, but I would like to present to you results that we obtained from experimental evolution of a microbiome member to this biphasic life cycle. And we chose a specific microbiome member, Pseudomonas lurida, strain MYB11, that we did find with C. elegans in the wild, but only occasionally. So we assumed that this is a microbiome member that is not optimally adapted to C. elegans yet, but shows potential. And then we ask, how does it change if we now force it into this biphasic life cycle where it needs to associate with C. elegans over time? And we did that over 10 cycles in 6 replicate populations. And this was work that was done, again, by 2 former group members, Nancy Obeng and Anna Czerwinski. We then analyzed the evolved bacteria from the end of the evolution experiment. We did it in various ways. We took entire populations, but we also obtained individual clones from the experiment, and we combined that with genomic and functional genetic analysis. So the first important result that we obtained is that the bacteria indeed improved their ability to associate with a host. This is what you see here, always on the left, and in red, these are the bacteria that evolved with a host in a biphasic life cycle. And then we had bacteria that evolved under exactly the same experimental conditions, but never saw a host. And here you see, and this is something we show in many other graphs as well, the result of competition experiments of evolved bacteria in comparison with ancestral bacteria. And this is then a measure for the ability of these bacteria to colonize the host very well and associate with the host very well. And you see a value above 1, so the evolved bacteria fare much better than they did before. And this is something that we particularly saw for the bacteria that evolved in the biphasic life cycle. A danger in this experiment is that in the end, we actually evolved pathogens. We checked that, and the evolved bacteria are actually not decreasing host fitness, and this is something that we measured here. So the ancestral host fitness or the fitness of the ancestral bacteria had been here at 1, and the evolved bacteria actually in both cases led to a higher host fitness. So what kind of adaptations did occur? And again, this is a very comprehensive data set, so I very briefly summarize it. The important observation we made is that under evolution to a biphasic life cycle, we identified a very specific colony morphology among the evolved bacteria. And this colony morphology is a wrinkly type. And this is a type that we only observed when the bacteria did evolve to host association. And it's a colony morphology type that we never found in any of the bacteria coming from the control conditions. So with this and some other data, we concluded that these wrinkly bacteria are the host specialists. They adapted. And they showed to be very well able to colonize the host. And then based on a multivariate PCA analysis, we could show that they are the bacteria that improved short-term persistence in the host and biofilm formation, and also release from the host in comparison to the ancestor. What made this even more interesting when we now looked at the genomics, irrespective of whether we did sequencing of whole populations or individual clones, is that only in these host specialists we found mutations that were related to activity of a second messenger that is very well known for microbiologists, cyclic DGMP. We never found any changes in these genes that influence cyclic DGMP in any of the other clones in any of the other populations. So this suggested to us that cyclic DGMP may be important for being able to adapt to the host. We validated this further by measuring cyclic DGMP for these bacteria. So here you have the ancestral bacteria, and then you have 3 different evolved bacteria. And then we measured cyclic DGMP activity in 2 different ways, with a fluorescence sensor and via mass spectrometry. And the darker the red color, the more we saw cyclic DGMP, and it's clearly a significant difference to the ancestor. We further evaluated the importance of cyclic DGMP and these mutations by performing further competition experiments, where we always looked for performance of a mutant versus the ancestor in the biphasic life cycle. And whenever you see a value above this dashed line, then the mutant performs better. And these are the clones that came out of the evolution experiment. And here the mutants perform better than the ancestor. If we then reconstituted the ancestral gene variants, then this advantage disappeared. So we performed genetic rescue. So we took away the mutations that we found in these genes, and then they did not differ from the wild type. And when we artificially introduced a mutation in these genes in the ancestral background, we could sort of reintroduce the advantage of these mutants. We furthermore introduced changes in these genes, particularly in this case in WSPF, in other strains of the same species or even other species of Pseudomonas. And in all cases, whenever we changed cyclic DGMP activity, they actually fared much better in host association. And to further generalize this finding, we looked at complete genome sequences available in GeneBank databases for all Pseudomonas species and looked for the presence of this WISP operon that is important for the regulation of cyclic GMP activity, and to what extent the presence of this operon or of single genes of this operon is linked to metadata highlighting that they are living in association with a host. And there's a clearly significant effect. So whenever these genes or the entire operon is present, then we have a significantly higher likelihood that these species or these isolates are associated with a host. So taking all this together, we conclude that cyclic DGMP is an important regulator of host association. So first of all, with our experiment, we could demonstrate that bacteria or occasional commensals can evolve host association relatively fast. And they do that by improving persistence in late phases of host association. And we did identify cyclic DGMP as the important regulator to mediate that. This is actually important on its own because it was already published before that cyclic DGMP is important for pathogens to associate with a host. But this is very likely a study bias because most people look at pathogens and study pathogens. And what we here found, and in the meanwhile, it's actually confirmed by other studies, is that cyclic DGMP is not only relevant for pathogens, but it is actually relevant for any kind of host association. So with this, I'd like to close the chapter on looking at microbes and how they can actually improve their association with a host in this interaction between hosts and microbes and hosts and the microbiome. And I'd like to move on to more complex conditions. So to really look at host microbiome interactions across evolution. So how does the microbiome affect evolutionary adaptation? It is something that you often hear and see in the literature, that the microbiome is important for adaptation. But then there are a lot of implicit assumptions that have not really been tested yet. So there's usually an implicit assumption that the host microbiome association is co-evolved and acts as a unit during evolution. And this is again often not true. People know it, but you still find this argument very widespread in the literature. And there's very little experimental data also challenging and seeing to what extent the microbiome really helps adaptation of the host. I should actually add, there is evidence that the microbiome can help the host, but there's more or less no data what this then means for the host. So are there additional changes in the host, or are there directly contributions coming from the host? So we thought that C. elegans is a wonderful system to study that. And to look at that, Carola Petersen and Inga Hammerich from my group, they actually established a mesocosm approach for experimental evolution. So essentially what we do, we chop up some vegetables, combine it with sterilized soil, put it into a container like this, and we actually do use the plastic boxes which had pipette tips in it before. And then we add some bacteria. Some bacteria may actually come in with chopped up fruits and vegetables. And then we add worms. And the worms are a genetically diverse nematode population that has been established by Hinrich that some of you may know. And that is actually used in the C. elegans community for a variety of different evolution experiments. And they come with the advantage that we have some baseline knowledge for this population. And in our case, we actually worked with this population before for when we studied host pathogen coevolution. And we are extremely proud that this experiment is now running for more than 4 years. We never expected that, but the worms just keep going under these conditions. And it's actually the first time that anybody maintained proliferating nematode populations for such a long time. This is how it looks like. This is the beginning, and then it proceeds like this, and this, and this. And to make it even more, or to illustrate it even more how it looks really, really ugly. And you may consider yourself lucky because you cannot smell how these compost mesocosms do. It's really worse than how it looks like. And the people who are working with them, so Carola and Inga, they always hope that the mesocosms will die out, that the nematodes die, so that they can finally get rid of the smell. Unfortunately, this is how it looks like. So if we take a little bit away of it, we always have masses of nematodes in there. So C. elegans seems to really, really like stinking sludge compost-like thing. And it seems to us that there really are populations proliferating under almost infinite population sizes. So we're very excited about that, very, very unexpected. It was more or less a chance effect that we tried the experiments like this. So there's now a variety of different things we can do with the experiment, and I show you one of the early analyses that we did. And then I will show you some, as a teaser, something that is currently ongoing. So we can do common garden experiments where we then try to assess to what extent the microbiome now helped C. elegans to adapt to these compost conditions. And it also may help us to understand to what extent the nematodes themselves changed, or whether it's both. So we took ancestral nematode populations, ancestral microbial communities, or adapted C. elegans populations, or acclimatized communities, and the other combinations as well. And what I show you now, this is a material that we obtained after only 100 days. And again, to cut a very long story short, I will only show you the most important results that came out of this experiment. So what you see here are the different combinations in the full factorial designs. So the adapted nematode populations combined with adapted microbial communities, the ancestral ones, and then the different kinds of combinations. And here we measured in the common garden experiment offspring per worm. And we measured it for one particular replicate that we found very interesting and promising. And the offspring per worm is measured under compost conditions, so the conditions that the nematodes experienced during the experiment. And what you can see here is that whenever the adapted nematodes were included, then we reached the highest level. And it looks a little bit like that in this combination where both are adapted, we obtain the highest values, even though this difference here, as you can see, is not significantly different. What made this then even more interesting is when we then moved this setup, the combination, the full factorial combination of all of these different samples to Petri dishes with agar, sort of the normal conditions under which we maintain C. elegans. And there the advantage that the nematodes had with their adapted microbiome completely disappeared. And this is suggesting to us that indeed it is the combination of both the changed adapted microbiome community and the adapted host population is important for adaptation to this novel compost environment. And this for us and the field is generally important because it was the first experimental demonstration that both can contribute. That it's not only the microbiome that can mediate adaptation to a novel environment, but it is both the host and the microbiome can do this jointly. We, in this particular study, then tried to understand what is responsible on the microbiome side to mediate this adaptation, and also on the host side. On the microbiome side, we stopped by looking, or we focused on looking at which microbe taxa are important. We have a little bit of functional information that in the end didn't yield much. But the minimum we actually did get out of it, that we could identify certain taxa, highlighted here, that are more common in the adapted environment, and that are likely, therefore, important for this adaptation. And what was very important for us in this analysis, that we did not only look at bacteria, we also looked at fungi. And the fungal analysis was always mirroring the analysis of the bacteria, which for me was very unexpected. And it also allowed us to identify certain fungal taxa, in this case, a Banatozyma fungus, that seemed to be important for adaptation to this novel environment. The host response was studied by using transcriptomic analysis. So my lab has quite comprehensive experience in transcriptomic analysis of C. elegans, and we thought that this will be the easiest way of looking, of trying to understand what kind of functions may be important for this adaptation, and also to assess to what extent actually something happened at all. And what you see here in this explorative PCA plot is, on the one hand, in gray, nematodes combined with the initial microbes, and in red, microbes coming from the adapted, well-adapted microbial community. And you see also initial worms in circles, and then adapted worms in triangles, which in this representation didn't change much. But what it highlights here is that the microbes do induce a very specific response. So for the transcriptomic analysis, we again had all different combinations of adapted microbes, adapted worms. And to again cut a very long story short, the combination of adapted worms, adapted microbes led to an increased stress response, and a very specific stress response where only certain stress pathways were upregulated. So our interpretation at the moment is that a change in stress response mediated the adaptation to this novel environment in interaction with the changing microbial community. And again, the complete analysis is, of course, much more complex, as we then illustrated in the paper. So to conclude this part, so here we could demonstrate that C. elegans and its microbiome do indeed adapt jointly to a novel environment. And again, this had not been shown before, that it's really that it can be a contribution of both the host and the associate microbiome. The current knowledge we have from this experiment is that the microbiome adapted by community composition changes. We did not yet look at evolutionary changes of individual microbial lineages, so something that Bosco is doing here with Drosophila. But it's something that is also on our list. And for the host, we're looking at increased stress resistance that was underlying the adaptation to this novel environment. Yeah, with this, I wanted to illustrate a little bit of the things that we have done and also published to look at host microbiome interactions and the evolution of these interactions. In the past few minutes, I'd like to give you a teaser of some ongoing work. So you can imagine that we are, of course, doing a lot of more analyses with these compost mesocosms. We're looking at the microbial communities in more detail, trying to understand how they change over longer time periods and what are important factors that shape these changes. But we're also, of course, interested in sort of getting a better understanding of what is happening on the host side. And here we're actually getting much closer to approaches and methods that you are using here as well to look at experimentally evolving Drosophila populations. So the questions that we want to address when looking at the evolving host population is to better understand how the host evolves really with an adapting microbiome. And we realize that there is a comprehensive knowledge gap in this field because we do not really understand what host genes can actually contribute to in a situation where you have a co-adapting microbiome to a novel environment. So which host genes contribute to joint adaptation process, and also the population genetic dynamics are unclear. And yeah, I think we do actually have a very nice situation to at least start an exploration of these processes and also an exploration of the possible underlying molecular mechanisms. So this is something we now initiated, a population genomic analysis of evolving C. elegans. We did this on pooled population samples from the compost mesocosm. And at the beginning, we had a genetically diverse population. And what is important for the analysis, this population was created by intercrosses of 14 genotypes that are all fully sequenced. So we have full genome information of the initial genotypes. And we now obtained samples from day 0, 100, 400, and 700. And 0, we had actually a larger number of replicates so that we also could see what kind of random variation there could be at the very beginning. What was also important for this analysis now is that we had 12 surviving populations and 4 populations that actually went extinct. So a small number of the compost mesocosms did actually go extinct. And we can now actually try to see whether they actually behave differently. So whether the changes of the populations of the genomes of the populations look different between the surviving populations and the extinct populations. Another thing that we did and didn't think much about while we were running the experiment, which we only realized later that this is actually really, really cool and helpful for any kind of subsequent interpretation, is that we had to introduce a change in the experimental setup in between transfer or days 100 and 400. We had to really seal the boxes to avoid any kind of mite or insect contamination getting into the system. And we only realized later that this actually led to a drop of oxygen in these containers. So we suddenly had more anaerobic conditions than before. So we obtained all these pooled population genome sums, and I'm sure you know these kinds of pipelines. They look very complicated. So I will not go into detail. And as I said, this is also really work that is still ongoing. We haven't really finished everything. I'd like to emphasize what helped us in the analysis of identifying SNPs that we then use for subsequent analysis, that we had full genome sequencing data of relatively good quality for the starting genotypes. So we, in the end, in our analysis, we only considered SNPs that were already present in these original genotypes, because there we knew that they must be there, that they are really reliable. So avoiding any kind of problems with artifactual SNPs that we could not properly verify. And this is, in my opinion, important, because then we are more confident that when we see changes in allele frequencies, that they are really real, even if the frequencies of the alleles are really, really rare. So this was SNP identification, and then we did some population genomic analysis with the identified SNPs. And again, this looks very complicated. I'd like to emphasize that we're using quite some similar approaches that are also used in Christian's lab. And we're actually aligning these with, based on advice from Nida Baghi, since she's now in Plön and not far away. And we're doing simulations to again confirm relevant SNPs, and using other kinds of statistical approaches as well. And in the end, what we want to do is, and we realized more recently that the haplotype-based analysis that was introduced here seemed to be very, very informative for us. So we are focusing a lot now on this reconstruction of haplotype blocks and then the analysis of these haplotype blocks. And to understand what kind of functions are selected, we are doing enrichment analyses. And there we are actually very happy and lucky that we have a C. elegans tailored database for enrichment analysis that we established based on all published gene expression data sets. And we realized already quite a couple of years ago that this is an extremely valuable resource. Because if you, what you usually do in enrichment analysis, you do a Go-term enrichment analysis, but this only allows you to identify functions that are conserved across organisms. Because Go-term databases, they will highlight functions that are conserved across many different species. And with that, you will actually miss some more specific functions that you only find in your organism. And the advantage in C. elegans is that there have been more than 2,000, a lot more than 2,000 gene expression studies done under a variety of different conditions or in a variety of different mutants, different contexts, and so on. And that is now our resource to do an enrichment analysis that is then gives us much more specific functions that any kind of genomic changes or transcriptomic changes may entail. And as said, this is all only work that is still ongoing. We have a couple of very interesting observations that we already could make, and I'd like to conclude my talk with these new observations. So one thing that we observed is that there's an independent spread of genotypes and alleles across the independent biological replicates, meaning we see relatively little parallel evolution. We do see parallel evolution, but in most cases, it's actually very diverse. And again, it's perhaps not surprising because here we have conditions with an adapting microbiome, and it's many microbes, and they can go in all sorts of directions due to chance. So we may even expect that there is less parallelism. This is highlighted in plots like this. So what you see here, this is an example for one of the C. elegans chromosomes. It's actually chromosome 1, only one of the evolving lines or populations. And then you see changes between 0 and day 100, day 100 to 400, 400 to 700. And whenever you see a small block here in a color, then that is a haplotype block that we inferred, and that shows a significant allele frequency change in this particular time window. And the color indicate the original genotype. So you can already see from this graph here that there are haplotype blocks from different original genotypes that showed significant allele frequency changes in the different time windows. We see different blocks changing across different time windows. And already here you see perhaps there's a little bit more happening in this second time window, which included the period where we closed the boxes and where we had a strong reduction of oxygen conditions. And this is actually confirmed if we now look at other evolving lines. And also if we then look across more chromosomes here, you can see very clearly that a lot more is happening during the second time window. And you can also see that there is a large variety of different haplotype blocks coming from different backgrounds that is sweeping independently across these chromosomes and the independent lines. And again, it's not black-white. So you also see something happening in the last time window or in the early time window. But overall, it seems that the music is really playing in the second time window. But for me, it's also very interesting to see that there is not a unique genotype or haplotype block coming from one background that is sweeping across all of these different independent lines, and also not even across a single genome. I already emphasized, I'm talking for too long, yeah? I already emphasized that there is the most interesting changes seem to be happening in the second time window, and this is an illustration of that. So here we look at one locus on chromosome 4 in only line 2. And here's the first time window, second time window, third time window, and you see the most dramatic change is actually happening in the second time window. Here it's again line 2, a different locus on chromosome 3, and a somewhat similar pattern that we see. It's not all looking like that, but in the majority of cases, we see the most dramatic changes happening in the middle time window. Last but not least, in the cases where we saw parallel changes, we then did an enrichment analysis using our taxon-tailored database for these analysis. And we could show then or infer that the genes that we find in these significantly changing regions are enriched for genes that are involved in resistance against abiotic stressors, and again, only certain abiotic stressors, resistance against pathogen, and we could also show that certain regulatory pathways, especially TGF-beta signaling, seems to be involved in these kinds of responses. And that is the teaser. We are very excited of what else we can find out. I discussed it with some of you. It is to some extent very confusing and a challenge to actually get some meaningful insights out of it. But we actually think that what I highlighted here is already very interesting, since it is one of the few times that we could actually look at host evolution while an co-adapting microbiome is changing as well. So final conclusion to this teaser again, independent evolutionary trajectories with some overlap in host microbiome evolution to novel conditions, and we see signatures of changes in stress resistance. And with that, I hope to be able to have shown to you that C. elegans is a wonderful system to study host microbe interactions in consideration of the ecology of the genetics, but also using the powerful tool of experimental evolution. Many thanks for your attention.